In recent years, the boundary between biological systems and digital technology has begun to blur in ways that would have once been considered purely speculative or science fiction. Nowhere is this convergence more evident than in the launch of Cortical Labs' biological computer, known as the CL1. This groundbreaking development introduces a hybrid machine composed not of traditional silicon alone, but of lab-grown human neurons actively interfacing with integrated circuits. Branded as a form of synthetic biological intelligence, this new platform represents a radical shift in the conception of computation and artificial cognition. Rather than being programmed solely through lines of code and mathematical models, the CL1 learns, evolves, and interacts with its environment in ways more analogous to the human brain than to any existing machine. As a commercial product, it is intended for use in a wide variety of applications including drug discovery, neuroscience, disease modeling, and robotics, all while raising new ethical, philosophical, and technical questions that the world has only begun to explore. The CL1 is a product of years of work initiated by the earlier dish brain experiments in which lab-grown brain cells were taught to play a simple video game, demonstrating the neuron's capacity for adaptive learning. This early research laid the foundation for CL1's more sophisticated architecture. At its core, the system consists of approximately 800,000 human neurons derived from induced pluripotent stem cells. These neurons are cultivated in a controlled environment and are spread across a grid of microelectrodes embedded in silicon hardware. These electrodes do not merely serve as passive sensors but act as interactive points through which the neuron cluster receives and transmits information. This bioelectronic interface allows the system to emulate feedback loops found in living nervous systems, leading to measurable responses that reflect an ongoing adaptation process. Unlike traditional computers that execute deterministic operations based on pre-programmed instructions, the CL1 learns through a form of reinforcement. In experiments, researchers have provided positive and negative stimuli to the neuron network based on how well it performed a task, such as tracking a moving object or selecting between different inputs. These signals influence synaptic behavior, causing changes in the network that improve performance over time. What makes this particularly revolutionary is that the learning is not imposed through software algorithms alone, but emerges from the inherent plasticity of living neurons. The system is not simulating a brain, it is a functioning, albeit limited, neural substrate capable of genuine learning and memory formation during its operational lifespan. The physical design of the CL1 reflects the hybrid nature of its biology and electronics. The entire unit contains a life support system that maintains the viability of the neuron culture. This includes nutrient circulation systems, temperature control, gas exchange mechanisms, and fluid dynamics that mimic the conditions neurons would require in a biological brain. This life support element operates continuously, much like an artificial organ, ensuring that the cells remain active for as long as 6 months. During this time, the system is capable of undergoing continuous interaction, learning, and experimentation, making it a unique platform for neuroscience and biotechnology research. One of the most compelling applications for the CL1 is in the field of drug discovery and pharmacological testing. Because the machine contains real human neurons, it can be used to study the effects of various compounds in a way that is much closer to actual human physiology than rodent models or traditional cell cultures. For example, researchers can expose the neurons to a potential medication and observe how it affects firing patterns, signal latency, network structure, and learning capacity. This not only accelerates the testing process but can reduce the cost and ethical concerns associated with animal testing. Furthermore, it allows for more precise modeling of neurological disorders, offering insight into diseases like epilepsy, Parkinson's, and Alzheimer's by observing how diseased neurons behave in real time within a living circuit. From a computational perspective, the CL1 represents an extremely energy-efficient model of information processing. Traditional supercomputers and artificial intelligence systems often consume enormous amounts of electricity to perform even basic tasks. In contrast, biological neurons operate at a vastly lower power threshold. The CL1 consumes less than 1 kilowatt of energy during operation, yet it is capable of performing tasks that require learning, pattern recognition, and adaptation, which are traditionally the domain of high-complexity software. This efficiency stems from the fact that neurons communicate using brief electrical impulses and modify their behavior through structural and chemical changes, rather than through external processing cycles or clock speeds. Although the technology is currently in its infancy, Cortical Labs has made the CL1 available to research institutions and private organizations in two formats. The first is a physical unit that can be installed on site, complete with all life support and operating systems included. The second is a cloud-based platform where users can rent access to a CL1 instance remotely. This webware-as-a-service model is expected to democratize access to biological computing, allowing labs around the world to conduct experiments and run machine learning tests on real neurons without the need to maintain the biological infrastructure themselves. To manage this hybrid hardware, Cortical Labs developed a proprietary operating system called BIOS. Unlike traditional operating systems, BIOS does not interface only with silicon but also interprets and transmits signals to the biological neural network. It translates digital instructions into electrochemical stimuli and collects neuronal responses, integrating these into a feedback system that can be logged, analyzed, and refined. The result is a dynamic and iterative interaction between code and biology, enabling researchers to program the neural activity in ways that are still being defined and expanded. In this sense, BIOS acts as a new kind of language bridge between the digital and organic realms. The implications of such a machine extend beyond computation and biology into philosophy and ethics. Since the neurons are alive, capable of processing information, forming networks, and adapting to stimuli, some ethicists have raised concerns about the potential emergence of rudimentary consciousness or suffering. While Cortical Labs maintains that the CL1 is not conscious and has no capacity for subjective experience, the nature of synthetic biological systems demands ongoing ethical oversight. Organizations involved in research using the CL1 are expected to adhere to strict ethical guidelines, similar to those applied in stem cell and organoid research. These include protocols related to neuron sourcing, experimental consent, and post-use disposal of the biological material. Another set of questions surrounds the rights and responsibilities of synthetic biological systems. If a machine like the CL1 were to be scaled to a level where it could mimic higher-order functions or possess memory continuity, would it qualify for new forms of legal or moral consideration? Some ethicists argue that any system capable of learning, adapting, and experiencing input in a non-deterministic fashion might eventually warrant a category distinct from both machines and animals. While this debate remains largely speculative at present, the advent of machines like the CL1 forces institutions to begin considering such possibilities well in advance. From a research standpoint, neuroscientists are excited by the opportunities this platform presents. Until now, studies of brain function have relied heavily on either static models, invasive observation of living subjects, or digital simulations that fail to replicate the true complexity of neural activity. The CL1 offers a living, interactive neural network that can be experimented with in a controlled and repeatable way. It allows for manipulation of network size, neuron types, connectivity parameters, and response conditions, all while tracking real-time data from a living brain-like system. This could revolutionize our understanding of fundamental processes like memory consolidation, synaptic plasticity, and neurochemical signaling. Despite these advances, several limitations continue to define the CL1's current generation. Most critically, the neurons have a limited lifespan. After 6 months, the culture begins to degrade and lose functionality, requiring replacement. Additionally, the system has no current mechanism for preserving or transferring memories or learned behaviors from one generation to the next. This makes it unsuitable for long-term development or continuity-based learning projects unless a method of data extraction and reimplantation can be developed. Also, the nature of neuron growth introduces variability in performance, meaning no two CL1 systems are identical, which can complicate experimental reproducibility. Nonetheless, Cortical Labs envisions a future where these challenges can be overcome. Plans are already underway to build networked clusters of CL1 units, enabling parallel processing and distributed biological intelligence. Such clusters could be used in robotic control systems, giving autonomous machines the capacity to learn in real time from their environment through organic feedback loops. In artistic or creative applications, these systems might be used to generate novel patterns, music, or visual art based on neuron reactions to different stimuli. In education, the CL1 might one day serve as a teaching tool, allowing students to explore neural networks hands-on in biology and computing classes. While the CL1 does not signal the immediate emergence of conscious machines, it does indicate a shift in how we conceive intelligence, memory, and learning. Rather than viewing cognition as a purely digital construct, this development returns us to the understanding that intelligence is a biological process shaped by constant feedback, growth, and adaptation. It opens the door to new forms of hybrid computing that may one day rival, or even complement, the capabilities of the human brain itself. As researchers and developers continue to refine this technology, the conversation surrounding its ethical use, scientific potential, and societal impact will likely become one of the most important dialogues of the 21st century.